We're happy to introduce Lynn Rothschild, who's here from NASA, and she's going to talk on Makers Beyond Earth, Synthetic Biology and Space Exploration.
Thank you very much. I feel totally inadequate going after the last talk. That was incredible, wasn't it? We have a slightly different problem at NASA. We're trying to get humans off this planet. It's sort of strange to me when people say, are we ever going to go to Mars? Are we ever going to settle elsewhere? Of course we will. The question really is it going to be within our lifetime? And that's one of the things that we've been working on, particularly the last 6 years in my lab. So I'd like to tell you a little bit about some of the things that we're up to right now. So I've been selling this program around the world, including places like Washington, and to people who are used to things with on-off switches, people who are not biologists. And so the way I try to suck them in is by saying, okay, now imagine you had this technology that had all these features. It was programmable, well, so is your cell phone at this point, and it's modular. Again, you can think of lots of things in that category. But say that it was also self-repairing, and it could do advanced chemical syntheses, including making polymers, and it had built-in solar cells, and maybe it could find as little as a single molecule, even when dormant, and on and on. In fact, it can go dormant without the use of petrochemical products, no energy input, so on and so forth, precise mineral extraction. It's evolvable, and guess what? We have 10 to 20 million variants available today. The idea is, they're supposed to get out their wallets and say, I want this now. And of course, the answer is what they're buying is life. Life does all of those things. So if you make that leap to starting to think of life as an enabling technology, you open up all sorts of new capabilities. So if you took life and you took these capabilities and on top of it start to bring in additive manufacturing, 3D printing, and maybe 4D printing, just imagine all the things you can do. So what I'd like to do for the rest of this time is really get your imagination going with the sort of things, again, that we're doing. So someday, we're going to be off of planet Earth, if not us, our descendants. And this is what Earth is going to look like from the surface of Mars. Well, actually, this was Apollo 8. This is from the surface of the moon. So let's go a little further. This is what Earth looks like from the surface of Mars. You see that little tiny dot? Everyone you have ever known, every place you have ever gone, every vacation you ever took, and so on, is all in that tiny little dot. And so to the settlers on Mars, that's going to be Earth, this little tiny dot, this data processing center in the sky, and maybe someday it'll be the stuff of legend, or maybe we'll have some kind of advanced propulsion system, so it's going to be, your weekend holidays. You got a 3-day weekend, you can go to the Earth. But that's what it's going to look like to people settling on Mars. The thing is that it's not trivial to settle there. So, for example, you need transportation to get there, and you want transportation once you're there. You're going to need some kind of habitat. You're going to need life support. I occasionally talk to people in the military, and they say, well, you think Mars is bad. We've got people up in the mountains above Afghanistan. And I say, yeah, that's tough, but at least you've got oxygen and gravity. We don't even have oxygen or gravity. We've got, basically nothing but this planet and what it's got in it. You need power, you need heat, you need light, and most of all, you need radiation protection because it's really nasty there. If you think we've got an ozone hole problem here, there's no ozone layer on Mars. But in addition, you have huge challenges. And for us, the challenge is up mass. So in other words, launching stuff against Earth's gravity. That's a huge one, and that's tied in with cost. It's very expensive to send these space missions. In fact, they say it costs about $10,000 to launch a can of Coke into low Earth orbit. And what do you have? You have a can of Coke in low Earth orbit. That doesn't get you a human settlement on Mars. You got to be able to store things once you're there. You got to be flexible. And you've got to have these things be reliable. You can't just call Amazon.com and say, oops, I forgot something, please send it tomorrow. You better be able to do with what you've got. It'll be the ultimate field trip. And so what we'd like to argue is that synthetic biology, as well as biology in general, is going to make this possible. So synthetic biology is taking the capabilities of living organisms, or perhaps making your own, and then tweaking them so that they're useful to humans. So just like we started doing synthetic chemistry about 150 years ago, we are now switching into an era where we're doing synthetic biology too. And so most of my colleagues are doing things like biofuels, pharmaceuticals, environmental concerns, sensors, and so on. And we're saying, why not do all those things on the moon and Mars as well? So if you do that, you start to think of taking the best and brightest from the biological world. So it'd be like if you could have the ears of a bat and the eyes of an owl, and you could run as fast as a leopard, wouldn't that be cool? Well, we can do that kind of thing for microbes and plants, maybe someday for humans, but right now we're focusing on small organisms that can then do what we want to for us by taking the best ideas around. So the idea that we're selling you is that you could someday do chemistry on Mars the same way we do chemistry on Earth using biology. Biology makes your yogurt and your alcohol and all sorts of other things. Why not take material from the Earth or maybe from your spacecraft and then repurpose that, have that changed into something useful like maybe your clothes or your carpet or your chair or your food or whatever. You could use organisms for material production, including making habitats, and then do all sorts of fancy things like embedding sensors, or maybe making alternate life forms. We use, for example, right-handed sugars and left-handed amino acids. Why not switch it around? Or why not even make electricity from living organisms? That sounds really bizarre, but everyone in this room is using electricity to drive their brain, and they're driving it from what they ate for lunch or the snack this afternoon. So organisms already can do these things. So let me show you a couple of proofs of concept of what we're talking about here. So let's start with biocomposites. So if you look at something really, really sort of basic, like a brick, and then you think about what you could do if you could make a brick, it's really incredible. You could build a biological laboratory, a canoe factory, a tattoo parlor, museums, all sorts of things. You get the idea. Boardwalks, chocolate factories, my daughter's here, so I have to throw that in, and a school in Africa, and maybe a very old school in Cambridge, and one in New Haven. In fact, New York City is mostly made of bricks. So you can see you could do a lot if you could get a brick on Mars. But how are you going to do that? Well, here's the raw material for bricks. Why not make bricks out of this surface material? The traditional way we make habitats for space exploration is we bring them. The Apollo missions brought a spacecraft. But now, for example, we take this material on the surface, we call regolith, and we make cement using biology. So now we've made a biological brick. And once we have that brick, we can make all these other things. So this is a very simple idea, and this is what a group of undergraduates from Brown University and Stanford University did in my lab in 2011. They were part of a big competitive synthetic biology program called the iGEM competition. I think we have one of the team members in the room right now. At least he said he'd be here. This is now the 5th year these students have been doing projects in my lab. And you can visit them over in the main science area. They're not actually all that far from the NASA booth. So we've done all sorts of cool things. You'll see a couple of the other proof of concepts. But this was something that was pioneered by this 2011 iGEM team making bricks. So again, a simple concept, taking biology, altering it a little bit so you can make concrete, and then selling the idea you can build buildings on Mars. Now let me give you another sort of biocomposite idea that we've been doing in my lab the last couple of years. And that is using biomaterials. So biomaterials are fabulous things. You think about it, we've got benches here that probably have some wood in them. I've got cotton on me. I'm sure people in the room with silk and wool and so on. You can use biomaterials to make glass structures, like you see the little diatom there, bones, trees. These are all biomaterials. We're composed of biomaterials. But no one is saying that we're going to take trees and fields of cotton and sheep with us to some place like Mars. But why not take these capabilities? And so what our solution is doing cellular printing, 3D printing with cellular organisms. So you put these capabilities in a cell, like a yeast cell, and you use those to make the wood or the cotton or whatever. And then you bring those capabilities with you rather than the sheep or the tree or whatever. And so you can make food or artificial organs or whatever on site. Now, some of you may be thinking, wait, we've heard some of this before. In fact, even maybe in the last talk, there are people who have gone out and printed ears and fingers and livers and so on. Here's some examples in Petri dishes. I got this off the web. We don't make livers and ears in my lab. But what we're talking about is something actually very different. What we're talking about is all these wonderful structural things that people make that you use on the Earth, but then taking those capabilities in a different form factor and rearranging it. So we could, for example, make a very skinny pencil with a bit in the middle that's maybe made out of a thin wire rod surrounded by a layer of silk, surrounded by wool, surrounded by the most perfect wood you have ever seen. We could literally do that. In fact, we're extremely close right now. We have the proof of concepts. What about biomining? We're going to need metals when we go to Mars. In fact, we need metals on the Earth. And if you think about it, the traditional ways of extracting metals use enormously heavy machinery. Or people think about bringing metals with them in a spacecraft. That's ridiculous because it's very heavy to launch things into space and it costs a lot. So what if you could engineer an organism that can go into the surface of Mars and be able to pull out gold or platinum, or even something like copper, one atom at a time, and then you could take these metals and reprint a cell phone or a spacecraft or whatever. And so some of the students from this 2012 iGEM team in my lab did exactly that. They call this the BioMining project, and what they did is engineer bacteria to actually bind copper on their little flagella. So this last picture here, the green parts show you where they're binding the copper. Very exciting stuff. So right now I have a graduate student in my lab from UC Santa Cruz who's specifically doing this for her thesis. The idea is you could take, again, your cell phone, I think I left it on the bench, cell phone, when it's dead, you could grind it up, pull the metals back out, and reprint. This is not sci-fi. This is stuff we're doing right now. Very cool, both on Earth, actually, and on Mars. Or as you start to look at exploration, there are other sorts of things you can do. So my iGEM team last year from Stanford, Brown, and Spelman did something that was a very cool concept. What they did is build a biodegradable drone. So the concept is that you need a body. And so what you see, that thing there that looks kind of like a dried sandwich been left on the bench all summer, that's a material that's made out of fungal hyphae. So fungi have hyphae that are sort of like roots for plants. And a company in New York, Ecovative, took a design that the students created, made a mold, and then grew the fungi in it and baked them. So they were nice and lightweight and hard. Then the students grew microbial cellulose, waterproofed it with genes from a wasp, put linkers on and all sorts of biosensors, and then in the end we added some commercial propellers just so that this could fly for the Discovery Channel. And that's what the prototype looked like. Very cool stuff, and this was done in 3 months. So this is again, not way, 50 or 100 years from now. These are the sort of things that we have the technology for today. Now, all of these concepts that I've been selling to you are great, but again, think, you're on Mars and there's only so much there. What do you have? Well, you've got some sunlight, you've got some carbon dioxide in the atmosphere, you've got some nitrogen, you've got some water. The water's not liquid, it's solid or in the atmosphere, but there is water there, and you've got some minerals on the surface. So how do you go from these raw materials to something that you could eat, or certainly a microbe could eat, and then produce something for you? And so the solution for that is, again, something that the 2011 iGEM team thought of, and that is a project that they called PowerCell. Now, the great thing is that here are the students looking young and enthusiastic, and I have to say that this was definitely the blind leading the blind 5 years ago, and they went out there and they won best presentation in the Americas, and they won the best new application in the world, so I'm exceptionally proud of this team. So what they did is come up with this power cell concept, which I'll explain in a minute. But what's particularly exciting about that is that in the next year or two, it ended up being selected for a payload on a German satellite mission, the Eucropus Mission. So the 2013 team worked on that a little bit more, and now we have some alums from the 2013 and the 2011 team who are working in my lab full time, getting ready to launch. And what's going to be very cool is the satellite's going to be able to spin at microgravity, lunar gravity, and Martian gravity, so we'll have some idea how these things are someday going to work on Mars and the Moon. So let me tell you what the concept is. This is what happens on the Earth. You've got the sun coming down on something like the grass. This is up in Bolivia. You've got carbon dioxide from the atmosphere. You've got water. And all these things go to feeding things like the plants. And the plants then make sugars. This is all sort of, high school, middle school biology. And then you've got the consumers, you've got cows and so on eating the grass, all very, straightforward. And from them we can get useful products like meat or milk or leather or wool or whatever. So what I'm saying is, why not swap out microbes for these different stages? So you now use a microbe to take the carbon dioxide and the nitrogen from the atmosphere and make sugars. You now have another microbe living off of those sugars and nitrogen compounds and so on, and that's what makes you those useful products, the wool or the wood or whatever, because we've engineered them to do that. And there's no reason we couldn't now take that ecosystem and move it off the Earth and someday to a place like Mars. So that's the concept, to say this satellite's supposed to launch in March 2017, so hopefully I can come back in a few years and tell you how cool those results are. But basically, in the meantime, this is what's going to happen. So here's sort of a cartoon of this cyanobacterium, this green stuff on the side. We'll be adding water, and it will start to photosynthesize. So it'll start to make sugars. The students from 2011 engineered these particular cells so they leak the sugar into the medium. Normally they wouldn't do that. They'd keep the sugar for themselves. So they're going to release the sugar, and then another bacterium, one that the 2013 team engineered, will change colors as it takes that sugar in and could then ultimately someday make wool or silk or whatever. But right now it's just changing colors for us. So let me leave you with this grand vision that someday when we go to Mars, we'll be able to, for example, take the sunlight and have that and the carbon dioxide that's there and the nitrogen and the water and the minerals feeding these production organisms that'll then make sugars and so on for other organisms that can make us our clothes, our chairs, our food, our fuel, our pharmaceuticals, our habitats, our bricks, everything, basically. So you've got these microbes doing all this work for us. And so with that, I will say ad astra, which means to the stars, and thank you very much for your attention.
Thank you, Lynn. We'd like to open up the floor for questions. If anybody has questions, please come on up to the mic.
Before anyone asks, no, I have not signed up for a one-way trip to Mars. A lot of my students have, but I have not.
Just kind of curious, as far as the resources, like you got those elements and things are there, but are they kind of limited in certain locations, like water might be more frozen at the poles and hard to extract? And then, the light is very limited, very weak, and there's productivity issues. How much, can you actually support and build? There seems like a lot of, boundary conditions on location and ability of these different resources.
Right, and what's worse is all the places we'd like to go, the alternative talk I would have given was searching for life in the universe. So whether you're searching for life or you're building a colony, it's never where the engineers want to go. They would like to go on some really boring flat place that's nowhere near. So you're right, so it's a compromise and question potentially of transporting. But there is water certainly in the atmosphere, and so there's potential for capturing some even in the mid-latitudes. But you're quite right. There are issues of where all these things are located.
What about sunlight? That seems like isn't it pretty weak over there?
It's weak, but you'd be surprised how little a lot of things need. When we're outside, it's about 10 to 100 times brighter than what our incubators are in the lab for growing things. And yet our eyes adjust so it seems just as bright in the lab, but I sit there with a meter and I know it's only a tenth or a hundredth as strong. So I think that this is all very doable. Yeah.
Yeah, so one of the things that I know that when we send stuff to Mars is we're very careful not to bring any new organisms there and pollute the environment. So what you're suggesting is to create new organisms and take them to Mars. So is there a conflict there, or is there something been thought of yet?
Again, you're getting sort of my split personality between being an astrobiologist and trying to put people there. You're absolutely right. And I feel very strongly that we need to do much more work with robotic missions well in advance, because I can't think of a much more exciting discovery for biology than finding a second example of life. And if we mess that up, that would be an enormous scientific travesty. So yes, we need to do much more on that in advance, there's no question. However, that was part of why I was suggesting building organisms with alternate biochemistries, like having a left-handed sugar and a right-handed amino acid, or tagging them in some way so that we know what is us and what might be a Martian. And just to finish that thought, a lot of people probably don't realize it, but NASA has a planetary protection officer, Cassie Conley, whose motto is all the planets all the time. Maybe it's a little Men in Black, but it really is a serious thing to look at contamination that we're taking forward and potential contamination back to the Earth. And so we're looking at that in the United States. We are a signatory to a COSPAR treaty, a Committee on Space Research, so many countries have signed on to these questions of planetary protection. So yes, it's something we're very well aware of, and I would be crushed if we destroyed it to the Martians. Yeah.
Hi. So I have a question about how, and I think your last slide, you mentioned that you're trying to recreate some of these materials that are naturally occurring on Earth, or synthesized by animals, to bring to Mars. It seems like the movement to bring life or bring humans to Mars is like trying to recreate, I guess, our history of, when we went from the Ice Age to the Stone Age to kind of the human path of invention, right? But is there any kind of, I don't know, I'm just thinking out loud here, but way of taking a different path of history, of adapting to the habitat on Mars? Is there any work being done?
The similarity that I see is it's a lot like our ancestors in the early colonizers of the Americas, whether you want to talk about Native Americans or the Europeans. In any case, no one brought all their belongings and food and everything for the rest of their lives and in perpetuity. Everyone learned to live off the land. And that's what our descendants on Mars are going to have to do. They're going to have to live off the land. And what we're trying to do is show ways that you can do that, taking the raw materials that are there. And the advantage the colonists had here is they had trees to chop down and there was air and all that. We're going to have to create all that. But I don't believe that other than that, in that concept, that we really are going to be recreating human history. Okay. That being said, I'm sort of sorry I took out a slide. If you think about it, bone, for example, and teeth are great building materials. Why don't we use bone and teeth today like the Neanderthals do? Because there's a huge yuck factor. But again, it's the sort of thing that we could make materials that take advantage of those components in the lab, sanitize it and use those for building materials. So maybe in a sort of vague way, it's recapitulating the Neanderthal huts, but not quite.
All right, thanks.
Hi, I just have a quick question because the organisms you are designing right now.
Oh, we're not designing organs. There are other companies doing that, though.
So the new debate, this new re-engineered, and I don't know how to call them, but they need a media, special media, for example, special food, and then they have evolved in the earth, for example, so they're used to live in the earth. How do you plan that they will behave once if you use them on Mars, for example, because they are not used to these new
Right, so
environment.
that's a huge question, and that's one of the reasons we're doing this Eucropus mission, to see, for example, what effect gravity is going to have on these production organisms. And we don't know, you know, in a way I'd be really happy if there was no effect, because then we could just start planning based on our laboratory experiments. But if there's a huge effect, we better know now. So you're absolutely right, we have to know how all these things are going to affect this.
I mean, have you think maybe using like bacteria that are like adapted to extreme environments, or like, I don't know what?
Well, I'm actually really glad you brought that up, because I've spent a lot of time doing work on life in extreme environments, and I didn't mention it, but our 2012 team came up with a project they called the Hell Cell Project. And that was making artificial extremophiles. So again, taking genes from organisms that live in boiling acid springs or in high salt or whatever, and putting them in another organism to giving them superpowers. That's why they called it Hell Cell. So yeah, and again, they were able to do an incredible amount in 3 or 4 months, and I now have a couple of PhD students working on that. So that idea of mixing and matching and taking the best out of the natural world is something that's very tractable now.
So what I'd like to do at this time is say thank you to you, Lynn, and she will have time for a couple more questions outside. So thanks so much.